So we're now going to finish with Tony Zador. So he's a professor at Cold Spring Harbor. I think Tony's had an unusual history, at least to me, in terms of his involvement in connectomics. I think he really started out as a systems neuroscientist and physiologist, and he has descended perhaps to molecular biology and connectomics, which is perhaps the opposite of many people's path. But Tony, I'm really looking forward to your talk, the third of these three. Over to you.
Can you hear me?
Yep.
Okay. So yes, in fact, before I became a systems physiologist, I was actually a computational neuroscientist and neural network researcher. So I've really come full circle. I just wanted to thank you guys for this fantastic opportunity and this great session. This slide basically summarizes a lot of what my lab has been doing for the last 10 plus years. We've been trying to figure out a way to grind up brains and convert them into neural circuits. And I'll say a little bit about that at the end. But a lot of my talk is going to be focused on sort of zoom out to 30,000 feet, or I guess given the international audience, 10,000 meters point of view, and ask why I think connectomes are important for AI. And I think the previous two talks have been great at sort of setting the stage for my more speculative talk. So actually one of the main things that drove me to become a neuroscientist was I watched a lot of science fiction as a kid. And these were some of my science fiction influences. This is actually a cultural test that sometimes I give, but these are, the question is, why can't we build these human level AI systems? Why aren't we there yet? And how do we get there? And when I started graduate school, the dominant paradigm was what's now referred to as good old fashioned AI. And the idea is that you start with those things, the most interesting problems in AI are those problems that are uniquely human, like high level cognition, playing chess, language. Then you move on to the somewhat so-called simpler problems like reasoning, which a handful of animals can seem to do, at least to some extent. And for the most part, you ignore the problems, the kinds of tasks that the vast majority of animals and that could be called sort of human or sapiocentric AI. So to push back on this, even 30 years ago, AI pioneer Hans Moravec once wrote, encoded in the large, highly evolved sensory and motor portions of the human brain is 1 billion years of experience about the nature of the world and how to survive in it. The deliberate process we call reasoning is, I believe, the thinnest veneer of human thought, effective only because it is supported by this much older and much more powerful, though usually unconscious sensory motor knowledge. We're all prodigious Olympians in perceptual and motor areas, so good that we make the difficult look easy. Abstract thought, though, is a new trick, perhaps less than 100,000 years old. We have not yet mastered it. It is not all that intrinsically difficult. It just seems so when we do it. And so in some sense, what he's saying is that what animals do is hard, cognition and reasoning are the easy part. And this has led to, I would say, this is leading to a version of the Copernican revolution, where, as you remember, Copernicus took our focus away from ourselves, putting the Earth, and really the important thing about the Earth was that we are on it, and toward the sun. And in the same sense, I think AI is now at the verge of a Moravecian revolution, where instead of focusing on humans as the most interesting topic for artificial intelligence, we're starting with the things that animals do, zoocentric AI, interacting with the world, which turns out to be extremely difficult for AI. And then moving on to the somewhat easier, once we have that foundation, we can move on to the somewhat easier types of problems like planning and reasoning. And then once we get these things as a solid foundation, the theory is that the things that are uniquely human, and there are some things that are uniquely human, such as crying out of emotion, we can move on to those, but with a solid foundation, we'll do it the right way. So our goal is to achieve mouse level AI. I think that's an achievable goal. And that mouse level AI would provide a solid foundation for moving on to human level AI. So the outline of the remaining part of my talk is, first of all, to describe how modern AI relies mostly on learning, whereas animals rely on innate structure, to talk about the fact that the innate structure arises from the genome, which specifies a wiring diagram, and that the wiring diagram is compressed through a genomic bottleneck, and then to talk a little bit about some technologies that my lab has been developing, MapSeq and BarSeq, which enable higher throughput anatomy based on DNA sequencing by converting the problem of connectomics into a problem of DNA sequencing. So as Grace already pointed out, AI really relies on learning. The typical paradigm in so-called supervised learning is to take a large data set of labeled pictures, maybe millions or tens of millions of pictures, and then use an algorithm called backpropagation to extract, to squeeze out the structure of that and put it into a synaptic weight matrix. And in these algorithms, typically the networks start tabula rasa. So we need huge amounts of labeled examples to learn all the parameters. But of course, that's not how humans work. Labeling objects every second for a year would be of only 3 times 10 to the 7 training examples. And for those of us who've had kids, it does seem like we're labeling objects every second for 3 years, but that's actually an overestimate. So this is clearly not how humans or any other animals do it. And this has been well recognized in the field. In fact, Jan LeCun famously said that if intelligence is the cake, the bulk of the cake is unsupervised learning, the icing on the cake is supervised learning, and the cherry on the cake is reinforcement learning. So he's saying, yeah, it's still all learning, just not the kind of learning that traditionally is used in artificial neural networks, namely supervised learning. These other two are not as data hungry. But it's clear that even unsupervised learning isn't enough. Squirrels learn to jump tree to tree pretty effectively in a couple million seconds of life. A colt can stand essentially at birth, and spiders can hunt at birth. And then there are some things that are complex behaviors that aren't learned at all, like termites build termite mounds, birds build characteristic nests, beavers build dams without instruction. And in fact, if you're interested in hearing or reading about or watching beavers build dams, I recommend the YouTube page Justin Beaver, the Rescue Beaver, where a woman adopted a young beaver who had lost its parents, and the beaver showed characteristically beaver-like behaviors in her home, as you can see from the damage the beaver did to the walls and his attempts to build a dam inside the bathtub. In fact, Hopi Hoekstra at Harvard studies what you might call this a similarly innate behavior. Burrowing in Paramiscus, there are 2 species that are very closely related, except one builds, the old field mice build a long escape tunnel, whereas the deer mice build a very short one. And it turns out through cross-fostering experiments that the kind of burrow they build is genetic, not learned. So you can take the pups of one and have them reared by the other, and they'll build the kind of tunnel from their biological parents. So it's very clear from an evolutionary perspective why we want to build in as much innate behavior as possible, which is that, you know, the moment a pup or any kind of a youngster is born, a clock is ticking, and the faster that creature can reach maturity and reproduce, the better off it will be, and the more likely it will be to survive to the point of reproduction. So the idea is that it's quite clear that you want to build in as much innate behavior as possible, not too much, because you want to retain room for flexibility to deal with the world. Evolution will typically maximize fitness at birth by incorporating innate structure. So basically, I'm pushing back at the picture that many people in the AI community have, which is the brain basically comes prepackaged. It doesn't arise mainly through learning. It comes prepackaged, and as an American, I will focus on this form of prepackaged cake mix. Okay, summary so far: AI relies on learning, and natural intelligence relies on innate strategies. So where does this innate structure come from? It comes from the genome. So basically, genes specify wiring diagrams. The innate behaviors are encoded somehow indirectly in the genome. So the genes specify the wiring diagram. So how many parameters does it take to specify a simple brain? We've already talked a little bit about C. elegans, which has 302 neurons and about 7,000 synapses. And in that case, there's really no challenge. The genome of C. elegans is about 100 million bits, and it's easy to specify the connection diagram of a 7,000 synapses arbitrarily in 100 million bits. No real challenge there. Every connection could, in principle, be explicitly specified in the genome. But for the human brain, it's a lot more complicated. Naive calculation says that, well, if we have of order 10 to the 11 neurons, here, I'll just go to the more complex or the more sophisticated calculation. Basically says that we have about 10 to the 15 bits per brain if you exploit the sparsity of human connectivity. But that's a far cry from the number of bits that we have available in the genome, even if we use every base pair to try to hand wire a connectome, we'd still be 6 orders of magnitude off. So it's obvious that the human genome couldn't possibly specify the detailed connections of a human brain. So this is well known. It's not really a mystery, but the fact is there's a genomic bottleneck. Whatever is specified about the human brain needs to pass through this genomic bottleneck, this compression, if you like, of the wiring diagram. And the reason, the way that the resolution of this mystery or this paradox is that you don't specify every connection in the brain synapse by synapse. Rather, there are rules, and these rules can be far more compact than the hand-wired specification. So in a C. elegans, you could, in principle, specify every connection, connection by connection. But for example, let's say that you had even a very large grid, it would take very few parameters to specify the simple rule that each neuron should be connected to its 4 neighbors. Now, that's not a very interesting circuit, but with more complex rules, we can apparently specify circuits that are interesting enough that they enable humans to do what we do and dogs to do what they do, like chew through my internet cable this morning. And so the actual Kolmogorov complexity of the wiring diagram of the brain at birth is much less than the apparent complexity. And biologists recognize this fact, and that's why we study neural development. So the summary of part 2 is that AI relies on learning from examples. Natural intelligence relies on a genomic compression of this W. And if I had more time to talk about this, I would go on to describe how the genomic compression actually is an algorithmic advantage because it forces evolution to squeeze out what's important, what it's extracting from the environment, and thereby provide better generalization. In other words, to enable the animal to interact with its environment in a more effective way than if it merely memorized everything that had ever happened to it. Okay, and there are 5 minutes. I'm going to talk a little bit about some technologies that my lab has been developing to enable a lower resolution and higher throughput method of assessing brain structure and wiring that's, I would say, complementary to the EM connectomic approaches that you've been hearing about, that you heard about earlier. And really, the idea of this approach is we'd like to be able to, in a single brain, in a repeated way, look at connectivity, and that's through a technique we call BARSI. Look at activity, let's say through a method like 2-photon microscopy, and then also look at gene expression within the very same neurons from which we've learned about connectivity and function. So what we'd really like to do, I'm not going to tell you about all this today, but what we'd really like to do is to be able to take an animal performing a task So my lab studies auditory processing. So an animal that's performing an auditory decision task to record a lot of the neurons in that. And then for each of the neurons whose activity we've recorded, to figure out its connectivity and gene expression to understand how it's embedded in the circuitry. So as we've already discussed, the challenge of circuitry is that the axons are all tangled together, as shown in this picture. And so it's very hard to trace any individual one unless you use a method like electron microscopy. There is another approach that was introduced about 15 years ago now by Jean Livet, Josh Sainz, and Jeff Lichtman. And there the idea is that we can trace axons in principle more easily if we could give them different colors. And they came up with some very clever genetic tricks to enable theoretical diversity of 200 different colors to be expressed in this bunch of neurons. The challenge of this technique, though, is that although in principle there are 200 colors, it's very hard to resolve that many colors. In fact, it's often hard to resolve more than 4 or 5 colors. So these 3 colors purple are likely different, but they appear to be the same. So our solution inspired to that is to replace colors with numbers. And that's the basic idea behind MapSeq. We unique DNA barcode inside of each neuron. And of course, the theoretical diversity there approaches 0 as the length of the barcodes become longer. And so very briefly, the technology developed by Ian Pycon, Eustace Kepschel, and several others in the lab was to make a library of barcoded viruses, each one expressing its own individual virus, and inject it into a part of the brain along with a carrier protein that drags the barcode out to the synapses. And that was really one of the more challenging parts of this, is to drag the RNA barcodes out to the axons and to the synapses where we need them. And so I'm just going to touch on a handful of the things that we can do with that. So if we really scale that up, Mengwen Huang showed that we can actually measure the projections of 100,000 or more neurons per brain using this technique. And here are the projections of 2 of these neurons out of that 100,000 that we got in this brain. The limitations of MapSeq, though, very briefly, are crude spatial resolution. We can't see much about the individual neurons, and we can't relate it back. So we can't relate it back to gene expression. We can't relate it to activity either. And so for that, we developed a next generation version of MapSeq called BarSeq, which involves in situ sequencing. And very briefly, the idea is to repeatedly image a neuron that's expressing this barcode, where for each round of imaging, just like in a sequencing machine, we can read out the barcode. And so here is a successive series of images of some neurons that have been barcoded. And you can see that the same neuron gets a different color for each round depending on the barcode in it. This can be used for axonal tracing in vivo, it can be combined with gene expression. And so in summary, AI relies mainly on learning, whereas animals rely on innate structure. Innate structure arises from the genome, which specifies the wiring diagram. The wiring diagram is compressed through a genomic bottleneck, and MapSeq and BarSeq represent a path to high throughput anatomy based on DNA sequencing. And I'm going to skip that and just do a plug for our NeurAI Scholars Program at Cold Spring Harbor, where we are looking for people with advanced training in artificial intelligence and embed them in a neuroscience group so that we can take insights from neuroscience and apply them to AI. And with that, I'll close with some words of wisdom that I always turn to when thinking about the brain. If the brain were so simple we could understand it, we would be so simple we couldn't. And I'll also thank my collaborators and my funders. And I went only 1 minute over. So thank you for your attention.
Thank you very much indeed, Tony, for that really exciting and wide-ranging talk.